Good evening and welcome. My name is Katie Kubina, and I will be moderating this expert panel to celebrate the 8th anniversary of the Northeast Canyons and Seamounts Marine National Monument. Designated as a marine protected area in 2016 by President Obama, the Northeast Monument is located about 130 miles off Cape Cod and spans an area of roughly the size of Connecticut. The monument features 3 undersea canyons and 4 seamounts that support a rich diversity and abundance of marine life. Co-managed by U.S. Fish and Wildlife and NOAA National Marine Fisheries, the monument represents the only highly protected area in the U.S. Atlantic and the only entanglement-free zone for migrating marine mammals, sea turtles, and seabirds along the entire eastern seaboard. Summer 2024 was full of discoveries. In June and again in September, New England Aquarium scientists conducted aerial marine mammal surveys, recording a dazzling array of whales and dolphins. In July, Mystic Aquarium's senior research scientist, Dr. Peter Auster, led an expedition on the RV Connecticut to study the surface and midwaters. Simultaneously, Dr. Martha Nizinski of NOAA's National Systematics Laboratory led an expedition on the NOAA ship Henry B. Bigelow to explore some of the deep sea areas of the monument. Never before has such an interdisciplinary science team been able to study and share a comprehensive top-to-bottom snapshot of this incredible place. Tonight, we have 6 amazing panelists who will be presenting, and we want this event to be interactive. So if you have a question, just put it in the chat, and we will get to as many of them as we can, both during and after the presentations. So let's get to it. Kicking us off is Dr. Peter Auster. Dr. Auster is a senior research scientist with Mystic Aquarium, as well as research professor emeritus with the University of Connecticut. Peter?
Thanks everyone for joining us tonight, celebrating the 8th anniversary of this Marine National Monument. As Katie noted, I was the chief scientist on this most recent expedition. On the research vessel Connecticut, which is a 90-foot seagoing laboratory designed to support modern ocean research in offshore environments. I've been working in the area that's now the monument on and off since 1984, so yeah, I'm old. Using mostly submersibles and remotely operated vehicles directed at studies of the seafloor and deep midwater communities. You're going to hear about the most recent seafloor work with Dr. Nizinski soon. And it was always clear there was lots of ecologically interesting things going on in the surface waters in this ocean wilderness, but time was always limited, and there was just not enough money to stay out longer or to bring more people aboard. But now, nearly 40 years later, with my colleagues on this expedition, people from Fish and Wildlife Service, NOAA Fisheries, Yale University, UConn, Mystic Aquarium, and with colleagues who were working ashore at Woods Hole Oceanographic Institution, we all agreed to focus on the surface waters, the epipelagic zone, to better understand the interactions, the diversity and the interactions of species like seabirds, fish, gelatinous zooplankton, those jelly animals, as well as using the latest techniques to sample environmental DNA to better account for those species that we don't see, but they leave traces of their presence. So with all this work over the time we had at sea, we used different sampling techniques to characterize the surface waters of the monument, allowing us to view nature through essentially through different lenses. We used baited remote underwater video cameras to literally bait or coax both large and small, highly mobile fishes to the video cameras, giving us video samples of species composition and diversity of species that are normally spread out and hard to sample otherwise. Indeed, we had limited time out in the monument because there were some ship issues and then some bad weather closing in on us. So we had a rapid 24-hour marathon of sampling over the top of Retriever Seamount, which is the seamount that's farthest away from the coast, about 1,800 meters to the summit. So we did blue water diving in the open waters. Yep, there I am. About 85 feet under the surface, and with about 1,800 meters, 25 meters under the surface and about 1,800 meters to the summit. So literally looking out into this ocean wilderness. We did that to look at mobile animals that associate with gelatinous zooplankton, and to be able to sample the gelatinous animals in individual jars so we can bring back fully undisturbed, undamaged specimens to Dr. Decker to be able to work within the laboratory. We also used a baited camera system that we hung on a wire, one of the winch wires from the ship. We lovingly called it Shark Cam, to be able to sample at night. And so that was just like the Bruce, but hung up to 600 feet below the surface of the ship. And while we didn't see jaws, we did see predatory siphonophores, large gelatinous organisms that have stinging tentacles to be able to prey on smaller planktonic organisms. We use sonar to locate the deep scattering layer, composed of diel vertical migrators, which is kind of a big word for the small animals like squid and shrimp and fishes that migrate up and down on the water column, up at night and down during the day to avoid predation. This is a synchronized movement of marine animals, again, between the ocean surface and deep layers, and it's widespread across all oceans and is considered one of the largest animal migrations on the planet. One of my colleagues, Yuan Liu from the NOAA Fisheries in the Milford Laboratory here in Connecticut, collected water at multiple depths using a Niskin rosette sampler with sensors, and we could see the sensor outputs live and trigger the individual water bottles at different depths. And when those returned to the surface, she filtered those samples to get DNA fragments to identify species that occur within and venture through the area to a depth of about 1,400 meters. Again, fish pee and poo and slough off mucus, all very nice things if you're a little boy, that leave DNA that can be used to identify who is there. Finally, we worked with colleagues at Woods Hole Oceanographic Institution using products from a NASA-funded study, taking satellite data that would let us visualize the sea surface landscape of ocean temperature, chlorophyll A, currents, and a variety of other oceanographic parameters that were used to predict the distribution of large oceanic predators like tunas and sharks. And we were able to communicate back and forth with a satellite system, so we could work essentially in real time with our colleagues back on the coast. I just want to close with saying how great it was to work with this group of incredible and collegial scientists, and the research technicians that helped us do the work, and the ship operators that kept us safe, and the ship's cook that fed us. In fact, that last is the keystone for any kind of expedition. And everyone made this expedition such an outstanding experience. Our plans are to get the band back together next summer and to bow to Neptune for more days of light winds and calm seas. You'll hear from several of my colleagues next, as well as from this expedition, and then venture into the deep sea using an ROV with Dr. Nozinski, and by air from the survey with Orla from New England Aquarium. So with that, back to you, Katie.
Thanks, Peter. Great stuff. And if you can't get to the monument in person, you can see behind me, there's a great exhibit at Mystic Aquarium. So we'd love to have you check it out there. It's sort of the visitor center for the monument. Next up, we have our colleagues from U.S. Fish and Wildlife. Maeda Miner is the Assistant Refuge Supervisor for the North Zone. She's the interim or acting superintendent for the Marine National Monument. And with her is presenting Dr. Mael Glon, who is the INM Supervisory Data Manager, also for U.S. Fish and Wildlife. So I'll turn it over to Maeda and Mael to tell you about their exciting bird surveys that they did.
Yeah, thank you so much, Katie. So we were fortunate in that even though our trip was a little bit limited, we were able to basically take advantage of all of the time that we had, both going out to the monument and once we were on site to do bird surveys. And so Maeta and I, our goals were to get out there and document as many seabirds as we could. There's not a lot of data about some of these offshore zones, and so really, every bird we were observing was potentially a novel data point. The way we did this, we primarily used a combination of binoculars, but also cameras. I found that using cameras could just be a really good way to get data, and it helps with identification. It helps confirm species that are unusual. We can kind of get some documentation for that. So we used a protocol that's the eBird pelagic protocol. So basically, we were out there and every hour was a different checklist, and we were up from, as soon as there was a little bit of light until we couldn't see anymore, and so we were on deck. Usually even having our meals on deck and just collecting data the whole time. We collected overall approximately 45 hours worth of data, so 45 1-hour-long checklists, and in total we saw 19 species of bird, which is a little bit low, but it was also a little bit quiet at some of the times, at some of the sites, I mean. Overall, though, we identified about 900 birds, so that was kind of an exciting number to have. And yeah, some of the more common species that we saw out there, we saw a lot of Wilson's and Leach's storm petrels. We noticed a really interesting shift in distribution where as soon as we get off the continental shelf, the Wilson storm petrels became less common and the Leach's storm petrels picked up. We also saw some differences in some of the shearwater species. So we had species kind of like this Corey shearwater closer to shore. And we also started getting some Sargasso shearwaters as we got further offshore. And a couple of sort of unusual observations that we had out there. One of the ones that was exciting was a least bittern, which is a marsh bird that should not be anywhere in the middle of the ocean, but that flew over our boat, so we were picking up some migrants. Meta was also excited to see a red-billed tropic bird that came in and circled our boat. And then we also had a white-faced storm petrel that off in the distance, so some of those were more unusual. Meta, do you have anything that you want to fill in?
Yeah, thanks, Mael. I guess I would just, echo what Mael had already said is, this was such a phenomenal opportunity. And even though the cruise was shortened, we had such a great window of, perfect weather while we were out there. So, Mael and I were pretty dedicated out there on deck. So the images make it seem like, it was action-packed, but like Mael said, it is many hours of just staring at the beautiful ocean and waiting to document which species are out there. So definitely a phenomenal experience. And, one of the highlights for me, other than seeing the red-billed tropic bird, which was phenomenal because that was at the top of my list, was just getting to work with all of the different scientists on board and getting to learn from them and all of the research going on in the monument.
And I also want to echo something Peter already touched on, but it was really great to have so many different scientists and to have people come up and just get excited about the birds we were seeing and sort of share information. I know some of the folks on the crew had been on other cruises like this, and they kind of told us about what they had seen. And then we were also very grateful for all the support staff who really allowed us to just basically eat, sleep, and bird, and we didn't have to worry about anything about the boat, anything about food. It was really just allowing us to do our job and do it as well as possible. And so yeah, definitely it takes a village to do these things, and we had a really great crew out there.
Absolutely. And as you can see from the videos, most of the time Mael and I have binoculars or a camera glued to our eyes. So definitely took advantage of the equipment we had on hand and just like Mael said, documented as many species as we could, and definitely looking forward to future opportunities to continue these surveys for pelagic seabirds, because this is really just a first glimpse into a much, bigger picture that we would like to understand with the species out and utilizing the monument.
Thank you, Meta and Mael. And one of the ancillary benefits to having two highly qualified individuals with cameras and binoculars glued to their eyeballs all the time was they also alerted us when there was spottings of whales and dolphins and flying fish, and took some amazing photos. So I think the entire group on the ship was very grateful for what they were able to offer us with seabirds and all of that glory, but with more as well. So thank you. Moving on, we are going from seabirds to the gelatinous things in the ocean, and I'd like to introduce Dr. Mary Beth Decker. Mary Beth is a research scientist within the Department of Ecology and Evolutionary Biology at Yale University, and just a delight to have on the ship with us. So go ahead, Mary Beth.
Thank you, Katie, and hello, everyone. As Katie mentioned, my role on the expedition to the monument was to survey the gelatinous zooplankton. And by gelatinous zooplankton, we mean the jellyfish and other fragile species that are out there in the monument and in pelagic waters. On the boat, I used a specialized net to collect these fragile animals. Here we are setting it up, getting ready to deploy. And as you can see, the end is a non-filtering cod end, which allows us to gently collect these fragile species so they don't get squished through the end of a straining cod end. And we're just getting this ready. And once it's ready, we send this down in through the epipelagic layer and then pull it back up slowly to gently collect the species that are present there in the upper layer. We also had an opportunity to use an underwater camera system. This allows us to, while the net can just give you a sense of what's in the whole water column, the underwater camera can show you where the animals are in the water column, at what depth they occur. It can also give you an idea of what other organisms the gelatinous species are present with. So it can give you an idea of what their prey might be, or maybe some of the predators on them as well. Here I am recording what we get in the net. So as the samples come in, I take them out and gently scoop them out. I will then put them in an aquarium. Here I am getting ready to scoop them out very gently with a dipper. And once I get them into an aquarium, I can then take pictures of them. And that's important because these fragile species don't stand up well to regular preservation techniques. So like this ctenophore that we're looking at here is just so fragile that once I put it into either ethanol or another preservative, it just falls apart. So at this point, I'm getting ready to photograph some of these species because this will help with species identification later. So this is Pelagia, which is a pelagic jellyfish that's on my background as well. Now you might ask, why study gelatinous zooplankton? Well, they're very cool and interesting and mysterious looking, but they also have important roles in the food web. They are consumers of smaller plankton like copepods and krill, but they're also prey for other species such as fish. Many species of fish eat jellies of various kinds. Also seabirds and sea turtles are known also to feed on jellies. And as Peter mentioned, I thought that was great how you mentioned that other species associate with jellies, and that is they use them as shelter. Just remember that picture from Peter's blue water diving. There's no place to hide in the open ocean, and so often some fish will hide under a larger jellyfish. So yeah, thanks. That's what I have for you today. I'd be happy to answer any questions. And also, I am grateful for the opportunity to work with such a great team, because I feel like we're all working together to understand all parts of the ecosystem. And it was such a great opportunity. Thank you.
Thanks, Mary Beth. And a good reminder for anyone, if they have any questions, to put them in the chat. I've been taking a look. And I know it's a small enough group that we can also get put ourselves on camera and audio at the end for questions as well. So we're moving away from the surface waters and into the deep sea. And for that, I'd like to introduce Dr. Martha Nizinski, who is a research zoologist from NOAA's National Systematics Laboratory.
Thanks, Katie. So as Katie mentioned, we're going to go deeper now and leave the realm of the midwater. I was the chief scientist of an international team that spent 14 days at sea investigating deep sea coral habitats found in submarine canyons off the northeast coast of the US and Canada, as well as some sites in the Gulf of Maine. We sailed on the NOAA ship, Henry Bigelow, that was packed to the gills with the Canadian remotely operated vehicle Ropos. And our first stop was Oceanographer Canyon. Oceanographer is the largest of 3 canyons in the monument, and it's characterized by high diversity and abundances of corals. The goal here was to survey an area that had been visited before because we have and this was a high priority because we have limited opportunities to do any kind of monitoring after areas have been designated as a protection zone. So the last survey in this specific spot was 20 years ago. So again, Oceanographer did not disappoint. If we can take a look at this amazing wall of bubblegum coral, we had the earlier view that was sort of zoomed out. So even with this zoomed out shot, it's hard to fathom the extent of this coral assembly. But as we zoom in here, you can notice that the bubblegum coral comes in 3 different colors. There's different sized colonies represented here. And notice how large the holdfast is. This is the location where the coral is attached to the canyon wall. And it has to be really big because it supports these large colonies, some of which were at least 5 feet tall. And you have these corals that are growing perpendicular to the canyon wall and in areas of strong currents. Corals also provide habitat for other organisms. And we were surprised to see how many brittle stars were calling this white bubblegum coral home. This yellow sea fan in the genus Paramaricia also occurs in high abundances. And next we can see that there are numerous colonies that were scattered over the canyon wall, but also notice that the geology has changed to a smoother, gentler slope. A high density of the Paramaricia was also observed on this outcrop. And I want you to notice too that there's this white bushy coral in the foreground in the genus Thorella, and this can also occur in high abundances in some places in the canyon. And in the next slide, you can see sort of, get an idea of how abundant it can be. But the real reason to show this slide is that in this very small area in this image, there's a high diversity of corals. And there's at least 6 different kinds of corals that are shown here. And some of them are very small size, which could indicate that new recruits are settling in Oceanographer Canyon. And that would be great news if that was the case. So our next stop was in Gilbert Canyon. And it's the least known of the 3 monument canyons. We visited this canyon in 2012 using a towed camera, and we're so excited to return with an ROV to gather more data and to collect samples in another unexplored area of the canyon. Gilbert is also characterized by high diversity of corals, with some occurring in high abundances. And here we see a field of that same yellow sea fan Paramaricia with a variety of other corals interspersed. We also frequently see large debris blocks, which are pieces of the canyon wall that have failed. The rocks are often clean of sediments and are ideal spots for corals to attach. And I love this picture because the corals are all lined up along the edge of this debris block, like they're guarding Gilbert Canyon. And if you look under the block, you get a glimpse of an octopus, and we saw many of octopus during our dives. It's also amazing to see how sheer the canyon wall can be in some places. And again, vertical walls are perfect places for corals to attach. Here we encountered another area of high diversity of corals, including the pink bubblegum coral, the yellow sea fan Paramaricia, some cup corals, white bamboo coral, as well as some anemones and sponges. Other places that we usually see corals are on edges of ledges, shown in the next slide, and under overhangs. So this bottle brush coral is actually a black coral, and it, along with the cup corals, were the most prominent corals in this particular area and in this image. But there are also some very interesting white sponges that are shown here. And these coral oases are always a great surprise, because here's a boulder that has fallen into an area with softer sediments, and it provides the only hard substrate in town for animals like corals to attach to. So there's at least 5 to 6 kinds of corals and a large vase sponge that are attached to this rock. So finally, to end this whirlwind tour of oceanographer and Gilbert Canyon, I have to finish with one of my favorite animals, which is this large squat lobster that was just hanging out on another type of bubblegum coral that we found in Gilbert Canyon. So back to you, Katie.
Thank you, Martha. So now that we've been to the deep sea, we're going to go way out and turn it over to Orla O'Brien. Orla is an associate scientist with the Anderson Cabot Center for Ocean Life at the New England Aquarium, and they've been conducting marine mammal surveys of the monument region for quite some time. So turning it over to Orla.
Thanks, Katie. Like Katie said, our team has been conducting surveys of the monument from the sky for quite some time. We've been flying since 2017, so within the first year that the monument was designated. And since then, we have flown 20 aerial surveys of the monument. So we've been doing a lot out there. So this is a photo of me just with our current plane. And we have a lot of good crew and observers with us that we rely on to help us cite all the exciting things that we see out there. So here's some of us suited up. We wear flight suits to fly around in the plane, some good safety gear. And have another photo of some crew. This was again before taking off for a survey. Probably happy thinking about what we're going to see. So when we get to the airport, we do a lot of setup, a lot of things that need to go into the plane, computers to take data on, a lot of different cameras. The plane needs to get fuel and whatnot. It's a successful survey is usually about 5 hours or so. I like this selfie because it shows you that, believe it or not, we all fit in this plane, and it's supposedly a 6-seater plane, not a 4-seater plane. So we all managed to cram in there. It takes about an hour to get out to the monument at our breakneck speed of usually 140 knots if we have a good tailwind, but then we slow down to 100 knots at our survey speed, because things pass by pretty quickly at that speed. So we need to kind of keep it slow and low to look out for sighting cues, to kind of look for disturbances in the water. And I like to show just a little video of, you're not going to see anything in this video, and then you'll know how we feel, because most of the time you're just kind of looking out and you don't see anything. You have to look for little splashes in the water. When you do see things, we come home and we have these like beautiful photos of all the whales and dolphins and things that we see. But what we're seeing inside the plane is still actually quite small from 1,000 feet away. So I like to give a little bit of an example. This is a group of sperm whales, and this is really actually pretty true to life of what it looks like for us when we're circling things. So just a good reminder of how hard we work to get those great photos. So I have a video here just of what it looks like to photograph out of the plane. We just have like a little pop-out window, and there was a lot of communication between pilots and so on, talking about, what we're seeing and how to circle. And then I think from here, I'm going to move on to a lot of the different stuff that we see. So I'm going to start off with large whales. We see baleen whales out there, like fin whales. This is a fin whale. They're coming here to feed on probably fish, small fish. We see little groups of fin whales, pairs, and so on. Definitely fun to see out there. In the monument, because of all the topography that you've seen in the last presentation, especially with all the rocky outcroppings and big walls, it's also, it's a great place for upwelling and for a lot of development of prey species. And one of the prey species that can be there is krill. And so we were surprised one day to see a blue whale shown here that came probably to feed on just really temporary aggregation of krill. That was really exciting. So just one day we saw 2 blue whales. But kind of on the other end of the spectrum, something else we see are beaked whales. Beaked whales are really monument specialists because they like to feed on squid. They're suction feeders. They'll go down and kind of slurp them into their mouths. These are True's beaked whales, and then I have some Cuvier's beaked whales a little later on. The 2 most common species that we see. The Cuviers in particular are really champion breath holders. They can hold their breath for, I want to say the record now at this point is over 2.5 hours, which is a pretty incredible feat. And that's a really nice photo of a Cuvier's from our survey in the spring that I really like. Something else that eats squid but is quite a bit bigger are sperm whales. They're definitely, I always like to say that it's not a surprise to see them, but you don't necessarily get to see them because they too can hold their breath for, an hour or so. So even though we might only see 1 or 2 on a flight, we know that there's dozens there, I'm sure, that we're just flying over. And that was a nice sighting last year of a sperm whale calf with an adult that might be the mother, might not be. And then I think from there, I'm going to move on to some dolphin sightings soon. So just another example of what things look like from the plane. This is a group of probably hundreds of dolphins. So I'm sure you're wondering how we see dolphins, if the whales look so small. Luckily, dolphins usually travel in pretty big groups. We like to take lots of photos of dolphins because there are many different species out there. So these are some spotted dolphins, usually a more southern species that come up. These are bottlenose dolphins. The watercolor and clarity out of the monument is really fantastic. So just enjoy some very peaceful Zen dolphins. Have some calves in here. Something else we see are striped dolphins. They're more of an oceanic species, so not something that you'd see close to shore. And we also saw, we see common dolphins a lot, and there was some really nice video from, I think, Peter's presentation of them bow riding. And they're always a fun treat to see as well. And I think maybe the last set of dolphins I have coming up are Risso's dolphins. Risso's dolphins and pilot whales I have that are kind of a fun set of species because they also are squid eaters, whereas most of the dolphins are kind of more fishy eaters. So pilot whales here travel in these pods or family groups. We often see a lot of calves out here. And they dive a little deeper than the Risso's dolphins, which are this pale white color. And so they can kind of like self-segregate themselves in terms of how they look for their prey and make sure they're not getting in each other's way. Something different from the cruises that you've heard a lot about. Obviously, we don't have videographers, so a lot of the, crew photos and whatnot are just us on our cell phones taking videos to share. We also don't have a chef, so it's observer prepared lunch. We don't go in to land as it takes an hour to get out there, so have a snack on the way. And then I wanted to end on a set of a few photos of some fishy things. So our goal primarily on all the different types of aerial surveys we fly are marine mammals, but we see just these incredible fish species, so we can't not document those as well. So we see these Chilean devil rays quite a bit. And we also see manta rays. That's a nice shot from a few years ago of a giant oceanic manta ray. And we also see sharks as well, more offshore sharks. So this is a hammerhead shark. We've started noticing those more recently. And then kind of the celebrity shark that we see is the whale shark. So these aren't necessarily unusual visitors, but they're just kind of, they only come up when the conditions are just right. So they like to come into this blue water that the Gulf Stream pushes up. So if you get the right conditions, you get whale sharks and all the different types of rays as well, filter feeding on lots of different things, maybe krill, fish larvae, etc. And I hope that has given everybody an overview of the huge amount of diversity and complexity that even just the surface of the monument holds. So, and I think that's all from me.
Thank you, Orla, and a round of applause for all of our panelists. We do have a few questions, and I will go through those. And as I am, if anyone else has a question, you can raise your hand in the react, the little heart button down at the bottom, and I will call on you, and you can put your video camera on and come off mute. So, but let me start with what we have in the chat here. So this is two for Orla. One of them was, what was the date of the marine mammal surveys that you're showing here?
So yeah, it's a great question. I just kind of did a collage of best of photos. So it's from like 20 surveys over the last 7 years or so. Generally, we try and fly quarterly. So we flew last week and we flew over the summer and in the spring. So it's most recent.
Great. And also for you, Orla, amazing. Can you talk a little bit more about what shifts you are seeing in marine mammal distribution in and out of the monument in the Northeast? I'm thinking about the orcas that are popping up.
Yeah, I think it's a great question. I think there definitely are marine mammal distribution shifts in the Northeast. With the monument surveys that we have done, because we do them quarterly, we're sort of slowly building up this database that maybe we could do that kind of analysis with that we haven't done right now. But in speaking to the orcas, for instance, that is probably just a result of, a little bit of dumb luck, maybe, that they're being spotted.
Great. Another question. This one is for Dr. Nazinski. How old would you say the bubblegum corals are that you were showing?
That's a great question, and we don't really know the answer. The skeleton of bubblegum corals is very spongy, and I'm not aware of any studies that have been done to actually age them. But given the size, we would say they're at least several hundred years old. Deep sea corals grow very slowly.
Thank you. Anyone else have any other questions? I'm looking through the participants, not seeing any hands raised, but if anyone does have a question, you're more than welcome to chime in with it.
Hi, Katie. This is Mary Camhigh. So, okay, I see Sarah. Let Sarah go, and then I'll add on to my questions.
You go ahead, Mary.
Well, I had a couple questions. So thank you, Sarah. First of all, this is really, really exciting. It's great to see, and it's a really wonderful overview. I have a couple questions just about one of them is, have you compiled the complete species list for what you've seen? It would be great to get a sense of that. The second thing is, I'd love to hear what you've learned from some of the eDNA work that has been done out there as it contributes to the species that you're seeing. And the last thing is, I'm just curious about what has changed. I know that I think you mentioned that there were surveys done at like 20 years ago, and now this is exciting to come back here and resample some of the same exact sites in the canyon. And can you say that you've noticed anything different that's notable? I mean, obviously, I know things change over time, but any noticeable differences?
Mary, was there anyone in particular that you were had your eyes on to answer those questions?
Well, I think the overall species list, I guess that would be just whether it was the expedition had compiled information that was available for the public. And, that's the same thing from whatever the eDNA survey work that was done. But anybody who has a sense about maybe those very early surveys compared to today, if we're actually, because one of the things we keep talking about is the need to do sentinel monitoring, long-term monitoring of these sites. And are we able, even at this time, that big time delay of maybe 20 years or something, seeing any major differences? So.
Well, knowing that Peter has spent the last 30 years studying this region, I'm going to turn it over to him to kick this off, and then maybe others would like to chime in.
Yeah, hi, Mary. So a couple of things. No, we haven't put together a full species list from the cruise yet because things are still being analyzed.
Okay.
And verified. The only people that have their homework done are Mael and Maida, because they got back to the dock with they put their data in a data set on eBird before we ever even got back to the dock. So that's really the way to do this. The eDNA work is still underway. The water samples were filtered, but then they need to get sent out for whatever that smart person stuff is that they do to be able to link the DNA to actual species. So that's still TBD. The deep sea coral stuff, and Martha, chime in I mean, we also had a survey out there in 2013. Right. And so there was work from 2000 and 2001 from Scott France and I were in Alvin and did transects up that wall, at least close by. And then there was the Deep Discoverer work from Okeanos in 2013. And then Dick Cooper had, and I don't have this in front of me, we've got Alvin still photos from transects, I think as far back as 1978. And in fact, I emailed Martha just yesterday saying, I got to go find those spreadsheets. I thought I had them on my computer here at home, but they're on a computer in the office, and I still need to get back to campus to dig that stuff out. So we can go back in time some number of decades to look at similarity and change in species composition. And indeed, from 2013 being the most recent at the time, we found the remarkable similarity over decades along that general area of the wall that Martha showed in Oceanographer Canyon. Although that wall full of bubblegum coral was pretty spectacular, and I don't think we've picked that up never in the past there.
Well, I will say that we had some navigation issues, so we were doing old school. We had to go old school. So hopefully we were in the right neighborhood and went over part of the same transect. But
well, so Paramaricia, the Paramaricia on the cliffs look very similar to what we had in the past.
Yeah.
And so we'll talk about the nav issues and see if we can figure out where all that was.
But of course the silver lining was we saw that great wall of bubble.
Yeah. It's not like you can call up Google Maps when you're out there.
No. And it does take a long time to analyze video data. Yeah. So like I say, we got back to the dock on the 31st of July, and so getting the video ready to put in the queue for the person who's going to annotate it. So it'll be several months to get through it all, and then to do the comparison with the historical ones. So you'll have to stay tuned, but we'll have an answer.
I will. Thank you.
Sure.
Yeah, and while we were discussing, Mael put the link to the eBird trip report in the chat if anyone's interested in seeing the results of the bird survey from the July expedition. And I know that if you go to the New England Aquarium blogs, they do talk about the diversity and abundance of the marine mammal species that they're surveying. So I'd like to, let me get to Sarah. Sarah, you had a question?
Hi, yes, everyone. This is so exciting. And thank you, Katie, for you and the others organizing this and sharing this with us. It's fantastic. I guess one question is, it kind of builds off some of what Mary was getting at. When do you expect the results to be summarized and distributed? Or is it likely that you're going to await the next segment of this adventure next year before, to collect more data before you try to publish anything or report on the results of this? So that was one question. Then after you answer that, I have a question for Martha.
Okay, so I think that's for Peter.
Yeah, I
Thanks.
guess so.
No, I'm just kidding. So I mean, we have a cruise report that we put together, but it doesn't have data results in it. And it's in all likelihood, unless there's some, well, I mean, this is probably going to be up to Fish and Wildlife and NOAA if they want to put some kind of results document on the web, because in all likelihood the results will end up in different places based on different topical areas. So everything doesn't coalesce into a single, necessarily coalesce into a single kind of paper. And with a shortened cruise that we had, ideally we go back out and do all this, at more places next summer. So there may be even a further delay. I mean, we certainly cut our teeth on how to do all these things in an efficient way. And it was some of this was a learning by doing exercise. And so we'll have to see what next summer brings. But like I said, we've got a cruise report that I've yet to post. There's a few last little edits to it, and so that's available to at least show what we did. But the results, other than the Seabird work, aren't there, at least quantitatively.
Great. Okay. And then for Martha, I did watch some of the live video stream while you were out there. It was so exciting. It was really
I'm glad to hear
amazing.
that.
I'm curious, where else did you go outside the monument? And can you speak at all to sort of the comparability between what you saw in the two canyons that you looked at in the monument and what you saw elsewhere?
Sure. Yes, so after we left Gilbert, we went to Hazen Canyon, which is one of my favorites. Again, another canyon that's just filled with large colonies of bubblegum corals. And Hazen is just to the south of the Hague Line. And then we went to Corsair Canyon on the Canadian side. And so the cruise that I was on is a transboundary cruise. And this was the fourth time that we've done this, where we, target canyons on both sides of the border, as well as places in the Gulf of Maine on both sides of the border. So we also did some work in Northeast Channel, which is a conservation area in Canadian waters. And my Canadian colleagues were very interested in going back there for the same reasons that we did this survey in an oceanographer, because again, they hadn't been to some of those areas for 20 years. So it was time to go back and look. And then we did some work in the Gulf of Maine on both sides of the border, again, looking just to find where deep sea coral habitats are, because we still don't know the extent of the coral distributions in that area. And particularly on the Canadian side, they're interested in finding these habitats to add them to their marine protection network. So we're trying to do the same on the US side, but Canada seems to have a faster pace of protecting some of these areas, but we're working on it.
Great. Thank you, Mary.
I see we have a question from Brad Sewell. Go ahead, Brad.
Hi, everyone. So my question is, I guess it's to Martha. How shallow do you generally get with the coral surveys? And then sort of related to that, are there opportunities to do more extensive work around the heads of the canyons, now that there aren't the same, like, fishing gear conflicts that might otherwise be present? And are there sort of novel opportunities available because the fishing gear conflicts aren't there?
That's a great question, and I know that's a question and an area of interest with the fishery management councils, is that, that some of the shallow areas, we know a lot about that. We know a lot about the deep areas, but it's that sort of in between areas that we don't have as many surveys. And we hope that there's going to be opportunity to try to fill that gap. But part of the reason, and part of the time we were out there, we're trying to think about ways that would be the most efficient to do that. And again, it's trying to find the right gear, and is a towed camera might be a better option, or an AUV that could do some moving around and get a good idea of what the habitat looks like in those canyon heads. And then that would give us more evidence of what's there, and if there are coral habitats of interest, then we could go back with an ROV and do some sampling. But I do agree with you that is a depth range that we don't have a lot of information, and part of it is because of fishing gear, and we have to be careful with ROVs and where we can put them in the water and how they maneuver around the gear.
Great question, and thank you, Martha. That brings us to the end of this webinar time. Once again, I want to thank all of the amazing panelists for pulling together to share their areas of expertise around this. It's so exciting what we are learning, and it's even more exciting what we hope to learn in the future. Sort of a once-in-a-lifetime opportunity to have this highly protected area and be able to study a place that's undisturbed. So thank you all, and thank you to all of the participants and the great questions, and we will certainly share this recording. So have a great evening and good night, everyone.